Good evening. Good evening, everyone. I want to express my appreciation for you guys coming out tonight. My goal tonight is to basically talk a little bit about this slide here. This is from suggestion from my wife because you can guess who I am in that picture. The idea that we work on the genome as a fundamental unit of information. Where is that information stored? How is it transmitted to the rest of the cell? And how is it transmitted to the rest of the other cells in the body or in an organism that's dealing with it? I've always been very interested in this idea because I grew up in a period of time when the genome was a real mystery. I was an undergraduate, and I remember doing work on RNA, and it was a molecule that nobody really knew what it did. We knew some RNA molecules did, but most of the time we didn't know what it was doing. And so when I decided to go to graduate school at the end, I wanted to understand this whole idea of where is this information in the genome. And so tonight I would like to talk to you about a fundamental thing in biology and genetics, namely the gene. What is a gene? But we're going to go at that question through sort of a backdoor way, through a personal experience. And the title here tells you about what was thought to be anathema, which totally improbable and unlikely to be true, and how that challenge actually led to insight into what a gene really is. All right, so that's my goal for tonight. So let me start by start with a little history. We're going to talk about the gene, all right, but it's the concept of a gene has been an evolving concept for almost 150 years. The first inkling of what it is comes from Friedrich Nietzsche, who was a biochemical physiologist, and he worked on white blood cells. And what he did was to basically take these cells and try to break them into parts, see what he could get. And one of the largest things you can get from a leukocyte is the nucleus. And so he isolated nuclei, and he saw this material in those nuclei. They didn't know what it was. They didn't know what it did. But he saw the same kind of material in almost every nuclei from every cell type. And he did some simple chemistry, and he knew that it was rich in nitrogen and rich in phosphate. And that's all he knew. Now, after his work, Kossel basically did some serious biochemistry on it, and he found out that it was a oligonucleotide. It was made up of long chains of these nucleotides, which they knew the chemical structure of. And this, because it was found in the nucleus and it was made out of deoxyribonucleic acid, he called it DNA. And that's where that term comes from. Now, that's over almost 150 years ago now, all right? But the next real insight into what a gene is came from these three gentlemen who won the Nobel Prize, Avery, McCarty, and McLeod, who basically showed through a series of very, at the time, elegant experiments that you could put DNA into an organism, right, and into a bacteria, and then infect an organism, and that infection would either kill the mouse or it would survive. And so they knew that this information that they were putting into a bacterial cell was information you could transfer from one organism to another and have a phenotype or a trait that you could actually monitor. So this was really the first real hard genetic evidence that information was in this material, which they knew was DNA. So this was the first inkling of that inheritance was transferred through DNA. When Jim Watson and Francis Crick worked out the structure of this DNA, not only was the structure defined, it was also how it could replicate. So how it could transfer that information from one cell to another, and how that these cells could grow and populate a whole organism, and how it could be transferred from one generation to another. So that was really important sets of information. But if you notice, all of this is physical information about the material DNA, all right? At the same time, roughly about 20 years, about 20 years after Kosovo, there was this botanist, Wilhelm Johansen, who worked on peas, just like Mendel did. And he found that you could transfer characteristics of peas, and he knew of the work of both Kostler and Meshner. He knew of that work, and he wondered, what is it in this DNA that actually could give these characteristics to these peas? And he figured out that there must be these kinds of units in this DNA, and that these units must be important because they could actually go and provide information to, as these gentlemen had seen, information that would lead to a certain phenotype. Now, this is a concept that he knew nothing. They knew nothing about DNA. They knew nothing about how this worked. But the concept was important because what he did was he called this unit a gene. All right, and it comes from the German, which means to go forward, to lead forward, as if you were passing things on. So he called it a gene. And this word is important to think of as not as a physical entity, because this will be an important take-home message here. Not as a physical entity, but as a concept. So how do you reconcile this physical element with a concept like this? This happened on a step-by-step basis. After Jim Watson and Francis worked out the structure, Francis came up with this idea that he could marry this idea of how replication could occur, how DNA could make copies of itself, and pass that information within the cell and to other generations by a molecule which actually we didn't know much about until the 1960s, believe it or not, called RNA. And that RNA later on was shown to be transcribed from the DNA, but they knew that they were together, and he hypothesized that the production of this RNA could then lead to the production of protein. So this became a central dogma through biology for a very long time. Now, so this leads us to this situation where we now have this concept of having elements, sort of units of what Johansen called genes, are scattered along the genome. And that's all we knew. That, in fact, we would call these sort of the bead string theory, that a gene just was present in the genome, and it would actually transfer whatever information was there to the rest of the cell, so a protein could be made. But the ability to have this happen gave certain implications. The implications were that a gene had discrete boundaries. It would have a beginning and an end. It would also mean that it would have a function, likely a very important function like make a protein. So these genes were thought to have only one function, pretty much, to make proteins. And finally, the other implication was that these genes, that function was carried out by basically being able to translate something in there. And since Watson and Crick had worked out this, what the nucleotide composition of this molecule was, there must be some kind of code in there that basically gave rise to these proteins. And that process we call translation. And ultimately, all that process led to was a trait. a phenotype. The question then became, what kinds of RNAs are made? How is this done? If everything was protein coding, all right, then it must be sort of a homogeneous collection of RNAs with different sequences on it. And that's basically what it was look like. But in fact, it turns out that with the advent of the human genome sequence being completed, there were methods by which we could actually go and look at where these quote unquote genes were, where they started, where they ended, and what the sequence was in them. And ask the question, what kinds of proteins were going to be made from there? So we, at the time, had access to a technology that allowed us to look across all of the RNAs in the genome, all right, simultaneously in a single experiment. And we could tell where the protein, what protein coding genes were being made in a given cell. And we did this experiment for a whole variety of reasons and cell types. And we found that most of the RNA that we found didn't code for a protein. It coded for something that could not make a protein of any reasonable size, but it was still made in a very discrete area in the genome, and it was made in abundance that rivaled what you see for protein coding genes. And here what you see is that today we know that there are roughly, 76,000 total protein and non-coding genes in the genome. And mostly 1/4 of which are only protein. But in fact, if you look in terms of the space on the genome, the protein coding genes only occupy 1.6% of the 3 billion base pairs. The rest is being, rest of the genome, up to 80% of the genome is actually encoding these non-coding RNAs. So what were these non-coding RNAs doing? So when we found this, and we published this in 2002 originally, and this caused immense uproar. People thought this was a waste of time. They were clearly noise in a biological system. And why are we wasting so much time and effort on things that we know aren't important? What's really important are these protein coding genes. So the problem then became how to find function for these. And I personally was very upset about it because I knew that these were being made just like protein coding genes. They were being made in response to given stimuli, and they were being made by very specific subsets at a very specific time. So they were being regulated in a very similar way, in a similar fashion. And this couldn't be just an accident, a wasteful product made by the cell. We and others subsequently thought about how to make experiments that actually brought function to this. And so what you see here is an example of one area that we found and other people have found of what these non-coding protein RNAs do. This gene here, called TP53, is the P53 gene. It's an oncogene, which means that it's if you mutate that gene, or everybody in this room has a P53 gene in every cell of your body. All right, you have 2 copies of it. All right, if you mutate that gene, most of the time you find it associated with a transformed state, a cancerous state. So mutations in this gene are critical in terms of establishing and maintaining that transformed state. That gene has lots of variations. There are 16 different RNAs made off of that gene. 16. And they differ by these little units, which you see depicted here, which are called exons. They're the coding part of the RNA. And they're not, so the RNA isn't translated as a string, it's translated as units that are brought together, they're spliced into a string that is that originally was distributed along the whole length of the RNA. So you can see there are 16 variants, not one variant, not just one RNA for each gene. There's 16 of them, and they differ based on the combination of these exons, these units that are in them. So you have a thing that was called a sort of a unit of identity, which is the gene, now having 16 components, each one of which can do something. Now, what do you mean you can do something? And what about these non-coding RNAs? So next door to this is another gene called WRAP53. It's a non-coding transcript and a coding transcript at the same time. It codes for a protein that basically extends the ends of your chromosomes and makes sure that they're extended. So they have a function as a protein to do that. But as an RNA molecule, you see this where the arrow is? This arrow overlaps the first exon of P53. That allows this RNA molecule, these two isoforms, these two versions of this that RNA, to regulate the levels of expression of P53. And so that, in fact, you can, in cancer, what happens is that becomes a regulatory element that allows cancer to happen because there are mutations in that first exon that now blocks that alignment to happen. So that's one of the functions that non-coding RNAs do. It regulates the level of transcription of either a neighboring gene or another gene in the genome, all right? Now, here's another example of what the function is, and also an example of the complexity. This on the top is two genes here, and you can see that these two genes are present at the top, and over it is overlapping a long non-coding RNA that spans all of these other genes. These are the ones in blue. That non-coding gene, we have no idea what it does. But it's overlapping exons exactly in every one of these protein coding genes. And we don't know how or why an RNA needs to actually occupy all of that space, share sequences with the genes that they overlap, and how that works. Now, down below, you see these two genes that are present. Right? There is another gene, another non-coding gene, right here. It is a regulator for this bottom gene here. And it does so not because it can actually bind to anything here. It does so at translation. When that gene is translated, it actually binds to the place where it's translated and regulates how much protein is being made. So it's a regulator. There are RNAs that regulate transcription, and then there are RNAs that regulate the translation of this. And there are many other functions. So the role of these thousands, literally tens of thousands of these non-coding transcripts that are being made in the genome all over, all right, are function to actually help regulate and guide the translation of protein coding genes. This is a remarkable thing. Nobody had the slightest bit of understanding of whether this was actually happening. And why would we need so many versions of a coding gene and so many versions of a non-coding transcript, right? That's because we have such a complex set of traits that are not just simple traits like color of eyes or color of hair, which require a lot of genes, but each one have different kinds of features, degrees, what we call penetrance, all right, in terms of carrying out its feature. And that is regulated in part by some of these non-coding transcripts, all right? Now, there is so much more I can say about it, but time is sort of limited. But I want you to have a sense that this was not easily accepted. And now it is more accepted in terms of its functionality and importance. But what happens is that at the end of the day, what was convincing to people was they saw it themselves. It wasn't just one lab, two labs. It's when people see this in their own work that they become actually convinced that maybe this is actually a reality. I'm going to finish with this last heresy that I wanted to introduce to you. And that is, we see this example about, I said to you, I want to talk to you about what a gene is. We see this example where we have coding genes, has lots of variants called isoforms, that combination, and we have genes that overlap them and share same sequences and share the same location and share regulation in the same fashion. Now, what do you call a gene? We call the gene usually just the one coding thing. But now we have all these variants that are being made and are being regulated in a very sophisticated fashion so that this kind of interaction can happen. Then what is a gene? Well, we think of genes as something in the DNA. But if the DNA is sharing its information among many different ways and in many different functions in the same region, the relationship between a locus and a trait, all right, has to be more complicated. And we see this as basically the role of the RNA. So the controversial thing, which is way far from being accepted, is that you might think of the gene as actually being the RNA instead DNA. Yes, DNA is important for the transmission and preservation of that information, but the function of what is actually going to happen to the trait comes from which RNAs are being made. So the controversy here, the sort of heresy that's being proposed, is that the unit of information in the cell is not the gene, it is the transcript, all right, the isoforms that are being made. And with that, I will leave you with that and take any questions that you have.